Okay, so we have this Punnett square, and what we're going to refer to here is we have the tall plant, which is crossed with a homozygous short plant. So heterozygous, so what heterozygous is going to represent, you're going to have make a square little Punnett square here like this on a paper, okay, just like that one there. Your heterozygous is going to be a big T, little t, and the homozygous for a short plant is going to be little t, a little t on the side there, okay? So down here in this first box, you're going to combine, you're going to have a big T, a little t. In this next box over here, you're going to have a little t, a little t. In this box down here, you're going to have a big T, little t again. And down here, you're going to have a little t again. So you are going to end up with a 50% heterozygous for tall plant and a 50% for homozygous short plant. Okay, and that's going to be your phenotypes and your probabilities there. All right, that is the same one. Oh no, so this one, the next time now, what you have is you have a heterozygous tall plant. Okay, crossed with a heterozygous. So you have a big T, little t, a big T, little t. So we're going to end up here as a big T, a big T, little t, a big T, little t, and a little t, little t. So what you end up with is 25% homozygous tall. Okay, up here in this corner right here, you end up with 50%. So this one and this one, again, is going to be heterozygous. And that's going to be heterozygous tall, and then you'll have 25% that is homozygous short. Okay. With incomplete dominance, this means that the phenotypes are not represented fully from the dominant and recessive genes. In this case, a lot of times both of the alleles are shown. So what we're going to talk about here is we're going to talk about red flowers and white flowers, and then the offsprings will then be pink. Okay, so that means a little bit of red is showing and also a little bit of white is showing. So here what we're going to have is we're going to have a homozygous red flower, and we will represent this with R's this time, okay? So we have a red flower with big R, big R. So this one is red originally, okay? And then over here we're going to have a homozygous white flower, again recessive, but this time it's not really recessive, okay? It's not really recessive, because they're all going to combine. And so you're going to have a big R, little r, a big R, little r, a big R, little r, a big R, little r. And that's going to give you 100% of them being, okay, that's going to give you 100% of them being pink because of the combination of them. Okay, so now we're going back to the sizing of plants. Okay, we're going back to the sizing of plants here. And again, we're talking about tall is going to be the dominant trait. And we're talking about medium plants here. And another medium plant, okay? So this is going to be heterozygous plants. We're talking about height now and also incomplete dominance. So that means that the heterozygous plant, that means that you have a big T and a little t, a big T and a little t, and you're combining them together. So you're going to get a big T, which is going to be tall again, okay? You're going to get a big T and a little t, which is going to be a medium plant. Okay, you're going to get a big T and a little t, which is going to be another medium plant. And then you're going to get a little t, which is going to be a short plant. So you're going to get 25% tall, 50% medium, and then 25% short. Okay, so that's going to be your genotypes up here in the box, your phenotypes, and your probability. Okay, these are the different classes of chromosomes you have here. So this is our human chromosomes that we have here. So we have 1 through 22, and then our 23rd set of chromosome is what determines our gender. So that here we have an X and a Y, and so that means that this is a male case of chromosomes. Okay, so now let's talk about the genetics of sex. In humans and other mammals, there are only 2 sex chromosomes. There's an X and a Y. If you have 2 X's, that will represent female. Fully in terms of genetics and sex, not in terms of gender. We're just talking about genetics and sex. If you have an X and a Y, then that will represent a male. Okay. So if you combine an X, so you combine an XY from, end up combining, so let's make this Punnett square really quick. Here's what we have. We have an X and a Y, which will be your dad, and you have an X and an X, which will be your mom. You have an X and a Y, an X, oh, sorry, that's an X and an X. Over here, you have an X and a Y. Over here, you have an X and an X, and over here, you have an X and a Y. So truly 50-50% chance that you're going to be a male or female. Okay. Now, some traits are sex-linked. So what that means is sex-linked traits are produced only by the X chromosome, and they can be dominant or they can be recessive. So what we see in genotypes with male and females that have sex-linked traits is that some of them do not express the trait. So an expressed trait typically can be seen on a male or a female who has the dominant trait. But even if a male has the trait, the female can mask the trait. Okay, so if it's a recessive trait in a male, then the female can mask it as well. Okay, now what about the genotypes for male and female that have sex-linked traits? Are they expressed? So it would be expressed if it was a recessive trait only on the male, and it would be expressed as for recessive trait if it's on both the male and the female. You wouldn't see it expressed if the male had any dominant trait at all, and you wouldn't see it expressed if the female had both dominant traits or only one dominant trait. Okay, most sex-linked traits are recessive. I believe one of these traits is going to be color blindness is a sex-linked trait. Okay. So we also have dihybrid crossing. This is one of the questions on your quiz here. So this is where you're going to make a Punnett square that now has 16 squares as opposed to the original 4 that we made. So a cross between 2 individuals that differ from each in regards to the 2 traits. So you're going to have their different pod color as well as their P color. So you have a fully dominant with the big G and the big Y, and then you have the fully recessive with the little g and little y. So your first generation is going to end up with big G, big Y, little y. And this, and then if you cross that again, let's see what you'd get. Okay. All right, so then for another dihybrid cross, we talk about here, this is all done out for us, which is really, really nice. So we have a homozygous tall with a green plant, which is TTGG, and is crossed with a white plant or a recessive plant, which is TT little g, little g. Okay, so you're going to end up with fully all across the board, all tall green plants, and nothing else is going to be tall and white or tall and short or short and white. Okay, and you see how they combine. So they take the first letter from the first one, and then a gene, and the second gene, and that's how they do it for that dominant crossing there. Okay, so polygenic inheritance is when the phenotypes are determined by the combination of effects of 2 or more genes at different times, the loci in the different chromosomes, and phenotypes are usually found in a bell-shaped curve. So these are the things that I was talking about, like hair color. When I was talking about it, it's actually polygenic inheritance. So it's not an exact science. Like if your mom has blonde hair and your dad has black hair, like you could have brown hair. Like that's totally a feasible thing. We typically see this in a lot of human traits that are physically like visible to us. So it comes out in skin color, it comes out in height, comes out in weight, comes out in intelligence, comes out in behaviors. So, you know, just because your mom's smart and, you know, maybe your dad didn't get the best grades in school, that doesn't, you know, fully mean that, you know, you're not going to get the best grades in school because your dad's genes were dominant or whatever. It's really on this bell-shaped curve. So you kind of take a little bit of both parents. It takes into account. Not to mention, we haven't even accounted for in here that your environment around you also affects this. So, you know, Jim Harbaugh, he talks about all the time how like everyone in his family is like just 6 foot or under. And he was like, no, you know, I'm not going to be that short. And he decided that he was going to drink milk every day. And he's convinced that that's what made him taller. I don't know. It's, you know, height's a little more based on genetics, but I'm sure that some of the other things that he did, you know, really truly eating healthy and eating well did help to contribute to, you know, how tall he is. So, you know, that's a big part of polygenic inheritance there. Okay. We also have multiple alleles that form in gene controlling of expressing trait. So in humans, we have this for blood types. So as you can see here, you can end up with multiple versions of type A, multiple versions of type B, one type of AB, and one type of O. Codominance is when a pattern of inheritance in which the phenotype affects two alleles in the heterozygous genotype express each phenotype of each allele fully and equally. Phenotype would be expressed in any other genetic combination. Okay, so like checkered chickens that have both black and white feathers. Okay, so both of them are expressed. So it doesn't, what this is saying is we had those plants where, you know, one was red and one was pink, or sorry, one was red and one was white, and they made pink flowers. Okay, so that was, you know, that was when, that was not codominance. I'm sorry, I'm losing my train of thought there. Anyways, so codominance on the other hand is when you have, like say you had a purple flower and a white flower and you combine them, they would be both purple and white. They wouldn't turn into like a lighter purple kind of flower. Okay, so that's codominance. Both of them are being expressed there. Again, here we go. We have environmental factors. So depending on what you're looking at here, so like these are both the same kind of fox, but they've adapted. So like this fox right here is going to be white, so he blends in with the snow, and that one's going to be brown so that he can fit in with the, you know, surrounding around him. Same thing, these flowers over here, they change color based on their soil or the sun. We change color based on how much time we spend in the sun. Okay, it changes our pigment and how much pigment that we have. Phenotype is controlled by both genes and the environment. Okay, we also have chromosomal mutations. This happens all the time. So it's damage to any chromosome due to physical or chemical disturbance or any errors during meiosis. Mutations might be inherited from sex cells of your parents. If the inherited mutation from your parents of all DNA will be made with the mutation already in it. The only way to inherit mutations from your parent, though, is through sex cells. So if they're on any other gene, then that's not going to happen. Okay. Skin cancer is not passed. Example, skin cancer is not passed on to your child. Okay. So mutations may cause certain types of diseases or make you susceptible to certain types of diseases. Two types of chromosomal mutations, the way it is structured and also the chromosomal number. Okay. So the two problems that we have with the chromosomal structure is the point mutation. A point mutation is where a simple change on one of the base of the gene sequence and its equivalent is changing one letter in the sequence. Okay, and then you have a frame shift mutation where a change in the bases that causes the sequence to be read in different sets or codons, equivalent to changing the spacing of a sentence. Okay, then you have gene mutations. I'll let you read through these. So you have a nonsense substitution where there's a change in one of the base pairs in the amino acid. You have a silent substitution where there's a change in one base that does not cause a change in the amino acid. We talked about amino acids before, but we'll talk a little more about them after mid-year. Some amino acids look exactly the same. And so that's why it doesn't, even though it is a change, it doesn't make any difference. Then you have a missense substitution where there's a change in base pair that also changes the amino acid that's produced. You can have a full deletion, which definitely changes everything. And then you can also have an insertion where you add a full, where you add something to it. Okay. Then we have chromosomal mutations. We can have a translocation where a portion of the chromosome breaks off and it attaches to a non-homologous chromosome. We also have an inversion where it literally flips upside down. And so then it's going to create different base pairs from there. Okay. You have a deletion, which we just talked about, where part of it's completely gone. And then you have a duplication where you're going to have one right after another. All of these are going to cause some sort of error in the mutation process. Some of them are fully visible when you look at humans. Some of them you can't even see. Some of them you don't even know, and they might come out later down the line. You also have a problem with the chromosomal numbers. So non-disjunction are members of homologous chromosomes that do not move apart during meiosis 1. Or sister chromosomes that do not separate during meiosis 2, which leaves one cell with few too many chromosomes and one cell with too many. This can lead to cell death, especially when we're talking cell death, especially when we're talking about like babies being born, which can lead to a miscarriage, just because we don't have enough chromosomes to match up there. So monosomy is the only one of the particular types of chromosomes. So you're going to take Tn-1. Trisomy is having 3 of one. So then you're going to take your normal number of chromosomes and you're going to add 1. And then polyploidy is having more than 2 sets of chromosomes. Triploids, 3N, 3 types of chromosomes. Tetra, 4N, 4 types of chromosomes, etc. So then from there, we get into some biotechnology. So this is where any procedure or methodology that uses biological systems or living organisms to develop and modify products for use. Specifically, genetically engineering the technology that can manipulate or alter genetic material of a cell. So what we're talking about here, that's a lot of words. I'm just going to skip to the next page. So we're talking about cloning. We cloned a sheep a couple years back in the 90s. So we know how to clone things. We're talking about, combining, sex cells in a test tube, and, searching and cycling for, certain hair color, certain sex. Those are all things that can be done. We can also do gene splicing. We do this with stem cells to help cancer patients. We take bone marrow and then we recreate them and turn them into new cells that can be used to help people who have cancer. This basically they become that part of their body becomes brand new with these new stem cells. So there's all sorts of things that can be used in these cloning and genetic engineering techniques. One, to help people, but also kind of just for scientific purposes to see, kind of how genes work. All right, and that's it. I know that is a ton of information. So this is, the final set of information for the semester. Please, please, please reach out if you guys have any questions. It's a lot of information. There aren't too many questions on your quiz. I really stuck to the kind of the Punnett square kind of questions. And then there's a few questions down here. Hey, cloning is one of your answers on your quiz. So keep that in mind when you're taking your quiz. But I really did stick to the Punnett squares. If you guys have any questions on that Punnett square worksheet, please reach out. It can be a little complicated, but it's really kind of simple, like once you get the hang of it. So please let me know if you guys have any questions again. All right, well, I hope you all have a great week, and I will see you later.